Hey everyone, welcome back to chapter 11. This is going to be part 2 of muscular tissue. In this particular section, we're going to explore a very important relationship, and that is going to be between the skeletal muscle and the nervous system. It turns out that one of the characteristics of a skeletal muscle, or one of the unique features of a skeletal muscle, is the fact that it's not able to contract unless it's first stimulated by a nerve. So that basically means that our nervous system, when we take a look at our neurons, our brain, our spinal cord, our nerves, they have full control over when our skeletal muscles are able to contract. So when the thin and the thick filaments are able to interact with each other, and you can see your muscle contraction as well as relaxation. And we see this importance of the relationship, especially if something happens that can sever communication between these two entities. So for instance, if a nerve connection is severed or even poisoned, we see that the muscle that it controls remains paralyzed, meaning that it cannot able to be contracted. And without using a muscle properly, we see that the muscle will start to become atrophy, meaning that it will start shrinking, the size will start diminishing. And this, of course, is very typical of the fact that the nerve is no longer able to use the muscle. And since the muscle is an organ, if it's not utilized, the body will shrivel it down because it doesn't want to maintain an entity that it no longer uses. So degenerative atrophy is a clinical application which basically says that you'll have shrinkage of a paralyzed muscle when the nerve remains disconnected. Now, I do want to kind of point something out as well. If you've ever broken your leg or your arm bones, I'm sorry, probably should be more specific than that, and you had to go into a hard cast, or you knew someone who had to be in a hard cast for a while. When the leg and the arm muscles are being immobilized on a temporary basis to allow healing of the bone, we do also see that those muscles will have slight shrinkage, meaning that when the hard casing is removed, the hand or the leg that was encased will be slightly smaller than the one that had full use and full range during the entire process. Now, this is going to be a temporary condition because the nerve and the muscles are still able to communicate. So once the patient gets rid of the cast and they go into their physical therapy and they start using that appendage again, you'll quickly see that the muscle will bulk itself up, and that's because it's able to communicate with the nerve, and the nerve will then send off the proper signals to get that muscle to contract. And the more we use the muscle, the bulkier it becomes, aka it will get rid of the shrinkage. If the nerve connection is severed on a permanent basis, like let's say for instance someone gets into a motorcycle accident and their spinal cord has damage and the nerves are severed, we see that once those connections are severed, the muscle will become paralyzed on a permanent basis. So that shrinkage will be something that will be on a full time and not be able to regenerate unless for some reason they could get the nerve connection to once again connect with the paralyzed muscle. Now, I do want to point out that the relationship between the muscle contraction and the nervous system is not only unique to skeletal muscle, but what sets it apart is that the skeletal muscle will solely depend on this relationship. Whereas when we take a look at our cardiac muscle and our smooth muscle, they too are able to respond to the nervous system in order to contract and relax, but they can also depend on other signals such as hormonal signals or electrical signals or ion signals. So they have a wide variety of options to get stimulated, where your skeletal muscle is solely based on this nervous connection. And that's why in this part, we're going to spend quite a bit of time exploring the relationship. And in a little bit, we're also going to take a look at what we call the neuromuscular junction, which is the exact meeting point between the nerve and the muscle and how they're able to communicate their excitement all the way down, leading to a contraction. But before we get that, let's talk about what we call a somatic motor unit. Somatic motor unit. All right, now these are going to be nerve cells, which cell bodies are in the brain stem and the spinal cord, and their whole mission will be to communicate with skeletal muscles. And in order to do that, we're going to see that their axons will extend out of the spinal cord and will make a direct connection with the skeletal muscle. So real quick reminder, based on chapter 5, when we were doing our histology, we saw that when you look at a neuron, and this is my best drawing of it, so bear with me. When we look at the neuron, we had the soma, the cell body in the middle. We had all the little dendrites that were able to receive information, and then they had one long axon. Here's the axon. This axon is going to be able to transmit and communicate with the skeletal muscle that it comes into contact with. And when you see that you have a neuron cell that's solely dedicated to the communication to your skeletal muscle, you can then go ahead and tell me that that is a somatic neuron. Somatic is going to retain the fact that it is referring to the skeletal muscles that it's serving. Now, a motor unit is now when you go ahead and specifically take a look at one nerve fiber and all the muscle fibers that it can innervate with. So go ahead and take a look at the illustration that is on the right-hand side, and you can see there's a little cross-section right here of your spinal cord, and what it's showing you are the fibers, the extensions of the axons that will leave the spinal cord and will go ahead and innervate or make a connection with the skeletal muscle. This will allow it to disperse throughout the muscle. This will also allow it that when the axon gets excited, it will allow the muscles that are in contact with the motor unit to contract in unison. So you'll be able to have contractions over a wider area. As long as the axon is showing its excitement and sending down its messages, the muscle cells will respond by contracting. And once that excitement goes away, then the muscle cells will then respond by relaxing. So what we see happening is that this relationship is extremely important to produce effective communication and ultimately lead to a controlled contraction and relaxation when properly stimulated to do so. There are different setups for our motor units. What that means is that there are different amounts of muscle fibers that each axon can communicate with or interact with. Sometimes they'll have just a fine amount, or I should say a small amount of muscle fibers that each neuron will interact with, and sometimes you'll have neurons that will interact with hundreds, if not thousands, of different muscle fibers. What this correlates to is what we call either a small motor unit or a large motor unit. A small motor unit, as defined on our PowerPoint, basically is when you have one neuron that's going to be in control of anywhere from 3 to 6 muscle fibers. And this is a small little batch of muscle cells, which means that when they respond and contract, it's going to allow for what we call fine control. So for instance, the fact that you can blink, or the fluttering of your eyelashes, or even your hand muscles, that you have fine control of when you want them to fully contract, half contract, how much strength you're putting into that contraction. That's all due to the fact that you have a full range of control of the contraction strength and the use of the muscles. So this will be correlated with a small motor unit. On the other hand, you can have a large motor unit, and a large motor unit usually means that you'll have your neuron that will be able to communicate with hundreds of fibers. And when these hundreds of fibers contract in unison, it's going to generate a strength or a very powerful contraction. In this case, the large motor unit is more for strength than it is going to be for control. So all the muscles, for instance, that are important for locomotion, walking, lifting, running, climbing the stairs. So like for instance, if we take a look at our calf muscle, our gastrocnemius muscles, our glutes, those are all going to be examples of large motor units because they have to be able to generate strength for the body to either propel forward for locomotion or, for instance, to lift any heavy entity from point A to point B. So a small motor unit is all about fine, delicate control, where a large motor unit is all about gathering strength. Now, the neuromuscular junction is going to be the connection point between the end part of an axon. so that's your neuron, and the starting of your muscle cell. And in this case, that's going to mean the sarcolemma. Remember, the sarcolemma is a specialized plasma membrane that you find in your muscle cells. So what we see happening here is that we're going to go ahead and take a look at a specialized synapse. So let's look at our PowerPoint. And right over here it says, a synapse by definition is when you have a meeting point between the muscle fiber and its target cell. Okay, so anytime you hear synapse, that's basically where the neuron communicates with its target cells. A synapse can be a wide variety of target cells. It can be one neuron discussing with another neuron. It can be a neuron discussing with a particular muscle cell. It can be a neuron with a specific organ. So what we're going to do is, to be specific when we talk about our skeletal muscle, we're going to go ahead and take a look at a specialized synapse called the neuromuscular junction. And the neuromuscular junction, whenever you hear that name, you're going to automatically know that is when my nerve is interacting with a skeletal muscle fiber. And in order to understand this, we're going to have to take a look at how it's anatomically set up. So what we're going to see, and I'm going to draw my axon again. So here are my little dendrites. And then here's my little axon. And as I said to you before, the axon is the one that will send the signal down, right? So your body has figured out that it wants a particular skeletal muscle to contract. So it's going to go ahead and activate the particular neuron, the somatic motor neuron that we require. And it's going to send that signal down the axon. And what we see happening is at the end of the axon, there will be a slight swelling. This is called the axon terminal. Okay, this is the swollen end of the axon, and it is going to hover ever so slightly over the sarcolemma. So this little line that I'm drawing right here, put a little arrow. This little line, this is my sarcolemma. So this is my plasma membrane of my muscle fiber, right? So what I see happening is that the axon terminal, the swollen end, will hover over this little dip. This little dip is called the synaptic cleft. It's going to allow the axon terminal to come extremely close to the sarcolemma, which is the start of our muscle cell, but they're not physically touching. They're literally just kind of hovering over each other. And this neuromuscular junction is extremely important, so we want to make sure that there is no outside entity, like a pathogen, coming in and destroying this relationship. So in order to do that, we see that there is something called a Schwann cell, which is an example of a neuroglial cell that will come and it will compensate and protect the neuromuscular junction. So it will isolate it from the rest so that there is no any type of pathogen or abnormalities that can portray and come into that particular location. All right, now I know my drawing was just perfect. You probably knew everything you needed to know from it. But in any case, let's compare that to what the book is trying to tell you. So here we see our axon terminal. And give me 1 second. Let me just click it. It's not responding to me for some reason. Come on. Give me 1 second. Where's my pointer? All right, here we go. Sorry about the delay. All right, so here we see our motor nerve fiber. And this is the axon. So we see that the axon is going to extend. And remember, we're going to have a knob at the end. So that's your axon terminal. And it's going to hover ever so closely to the sarcolemma. Here's my sarcolemma. That's my plasma membrane for my muscle cell. And you can see they're not physically touching. There is a space, your synaptic cleft right here. And then the Schwann cell is basically what's protecting any type of outer entities from coming to the inside. Now, when you're looking at this illustration, I'll make it a little bit easier by erasing some of these markings. You're going to see a few of the characteristics that we're going to have to discuss in order to fully understand this relationship. And don't worry, we're going to see this in the text coming up, but I just kind of want to show you the players, and then we're going to go over to the next slide, review it, and then we'll come right back to the picture, okay? So the first thing I want you to notice is that when you take a look at your axon terminal, notice how on the inside you have these little green little circles that have been drawn for you. And these are going to represent synaptic vesicles. So they're little plasma membrane, little containers, and they are going to be filled with acetylcholine, ACH. Acetylcholine is a neurotransmitter. And what we're going to see happening in a little bit is that when the axon is sending down its message that it wants the muscle to contract, the way it's going to communicate that with the muscle is that when its neurotransmitter gets down to the axon terminal, it's going to release these synaptic vesicles. It's going to do exocytosis. So it's going to release these little green structures into the synaptic cleft. And as it's releasing it, it's going to release the acetylcholine. The acetylcholine is going to be your neurotransmitter. It's going to be a chemical messenger. And it's going to travel over to the sarcolemma. And if you look at the sarcolemma, you're going to notice that it has these little blue little lines all over the place. And they're labeled as acetylcholine receptors. So the acetylcholine that's being released by the axon is going to travel over to these receptors. And that is how it's going to be able to establish a direct communication between the axon and the muscle cell itself. The other thing I want to show you is that notice when you look at the acetylcholine receptors and you look at the sarcolemma, notice how in the synaptic cleft, the sarcolemma has these like folds. These like finger-like folds that it's making. And it turns out that these folds are called junctional folds right over here. And their purpose is to increase the surface area. By folding inwards, it creates a larger space for the sarcolemma to add even more acetylcholine receptors. And the more receptors you have, the quicker you can respond to the acetylcholine that's being released from the axons. Now, I understand that some of you are still trying to kind of piece together how this all goes. So I just want to show you one thing on the picture. Notice how on the top part of the picture, it's all about the axon, right? And then the bottom part is all about your muscle cell. And it's trying to show you that on top right here, you have your communication between your axon and your muscle cell. But look what they drew for you all the way at the bottom of the illustration. Those are our good old myofilaments that we discussed in part 1. So those are your thin filaments, your thick filaments, your C disc, your sarcomere, the contractile unit of the muscle. So what we're trying to do is we're trying to get those myofilaments to interact with each other so we can get the muscle to contract. But in order to do so, the message has to come from an axon. So that's where your neuromuscular junction is so important, because it's going to allow you to establish a connection point between the axon and the muscle. And once the muscle communicates with the axon, it's going to do so on the sarcolemma level, so on the outside level. And we're going to eventually have to take that message and get it to come inside the muscle cell so we can communicate that with the myofilaments. So this is how all the different pieces start fitting together. Let's take a look at our next slide, and let's take a look at the written portion, and we'll come back to the picture to review, make sure that everything is covered. All right, so it says right here, it says the nerve impulse coming down the axon will release the synaptic vesicles. Those are those little green structures we saw in the knob, and it's going to release acetylcholine into the synaptic cleft. Remember, that's the opening between the axon and the sarcolemma. The synaptic cleft will allow the acetylcholine to travel over to their receptors. These receptors are embedded within the sarcolemma. And the sarcolemma, in order to increase its surface area so it can accommodate more receptors, will have junctional folds. And in fact, we have seen that there are disease models, such as myasthenia gravis, where when individuals start to lose the receptors or they lack the receptors, that the muscle will be very weak in its contraction. And if all the receptors are lost, then there will be no muscle contraction at all, because once again, you start to lose that connection between the nervous system and the skeletal muscle that is needed for the contraction to occur. Now, I also want to tell you something. When we go back to the picture again, I'm going to show you the basal lamina. The basal lamina is a collagen and glycoprotein layer that is separating our Schwann cell and our muscle cell from the surrounding tissue. So it's like an extra barrier that's going to isolate the neuromuscular junction. And the basal lamina is extremely important because it turns out it's going to allow us to have access to acetylcholine esterase. ASE means it's an enzyme, and the acetylcholine esterase, its main function is going to be to break down acetylcholine. Now, some of you might be wondering, wait, I need acetylcholine to communicate between the axon and the muscle so the muscle knows that I need it to contract. So why do I need an enzyme or acetylcholine esterase to break down my acetylcholine? Well, very simple. There will come a time where you want your muscle to relax. And in order to speed up the process and get your muscle to relax faster, we need to sever that relationship between the nervous system and the sarcolemma, obviously. Part of that will be to shut down the nerve impulse coming down the axon. But you might still have some acetylcholine floating around in that synaptic cleft. And if you don't want that acetylcholine to get to the receptors and stimulate the muscle any further, acetylcholine esterase, that enzyme, is extremely handy in getting rid of any excess acetylcholine that's no longer needed. So in a natural setting, acetylcholine esterase, it will break down acetylcholine in the case for your muscle relaxation to successfully occur. All right, so here's our picture again. So just to make sure, even if you have to pause the video, make sure that you're able to explain the function of the axon terminal. That's where the nerve signal is going to come to an end. Be able to explain that that's where you're going to find your synaptic vesicles full of acetylcholine, that the acetylcholine is going to be released into the synaptic cleft, that the axon terminal and the sarcolemma of the muscle cell do not physically connect to each other. They simply hover over each other. They are protected by the Schwann cell as well as the basal lamina. And coming back to the synaptic cleft, be able to explain that it has the acetylcholine receptors and also that it has the junctional folds, which are going to increase the surface area so you can accommodate more receptors. Oh, and I almost forgot that the basal lamina is going to be your source for acetylcholine esterase. That's going to be the enzyme that breaks down acetylcholine. So take a second, review all these different parts, because the neuromuscular junction is a very important entity to understand, and it's going to make it easier for us to explain all the steps involved in getting your muscle to contract if we understand who all the key players are going to be. All right, so let's talk about the 4 phases that are needed for your skeletal muscles to contract and to relax. And this, of course, is going to involve full cooperation of your nervous system. So the 4 steps are going to be excitation, excitation-contraction coupling, contraction, so this is when your muscle actually responds to it, and then, of course, all good things must come to an end, relaxation. Excitation is going to be all about getting your nerve to send a nerve signal down its axon and releasing that acetylcholine. The excitation contraction is going to be once the excitement reaches the sarcolemma, now we need to take those acetylcholine receptors and be able to transmit that communication between the nerve and the muscle. We need to be able to take that excitement and spread it from the sarcolemma all the way deep into the muscle cells where we're going to find our myofilaments. So these are our thin and our thick filaments. Because remember, They are the ones that need to make a physical contact. They are the ones that have the sarcomere, from one Z disc to the next. They are the contractile units of the muscle itself. Now, contraction step 3 is going to be all about making your thin and your thick filament physically connect and do what we call a power stroke, which is glide over each other, resulting in the muscle contracting. When this is all said and done, the 4th step will be relaxation, which means that we're going to need to, 1, get the thin and the thick filaments to no longer communicate, no longer connect with each other. We're going to make sure that the stimulus from the nerve stops, so no more acetylcholine is released, and that the muscle will then go back to what we call its resting length, what it normally will be at when it's not contracted. Now, here's where the fun part begins. We're going to explore each of these 4 steps in detail. So we're going to do this through an illustrated example, and I'm going to walk you through all the different steps that are involved. So I'm very excited. This is one of my favorite topics in A&P, so I hope I can share my excitement with you. All right. All right, so let's take a look at the first step, which is excitation. Now, we know that this is already going to involve the nervous system because the nervous system is the one that's going to regulate contraction of our skeletal muscle. So our first step for excitation is the fact that we're going to have a nerve signal that's going to travel down the axon and make its way over to the axon terminal. When it does so, one thing that's really important to point out is that when the nerve signal enters the axon terminal, we're going to also need calcium to enter the axon terminal. Make a little notation for yourself right here. Try to draw a little star. This is a very important step. So when the nerve signal comes down the axon, you are going to need some calcium to enter into the axon terminal as well. All right? Now, once the nerve signal makes it to the axon terminal and you have your calcium that has entered, the second step is that our little synaptic vesicles, our little green vesicles, are going to do exocytosis, and they're going to release acetylcholine into our synaptic cleft. And the acetylcholine is going over to the acetylcholine receptors. So here, I'm going to circle it for you. You have a little maximized image or an enlarged image of what a little receptor looks like. And these little green dots, those are my acetylcholine proteins that are meshing with it, or ligands, I should say, not proteins. All right, so step 1, arrival of the nerve signal. We need calcium to make that successful. Step 2, acetylcholine is released into the synaptic cleft and starts to connect with the acetylcholine receptors. All right, now once the acetylcholine matches with the receptors, we're going to see a response to that receptor by the receptor opening up its ligand-regulated gate. Ligand-regulated gate. So important to point out ligand. Ligand means that you rely on a solute or a particle for that gate to open up. So in this case, you rely on acetylcholine. So acetylcholine needs to be attached to the receptor. And once it's attached, it will go ahead and respond by opening up the gate. And this will allow sodium to come into the sarcolemma. See, here's my little sarcolemma. And potassium to be kicked out of the sarcolemma. And we're doing this because, as you might recall, muscle cells are excitable. You can change their membrane potential. So very similar to a battery, you can change the membrane voltage that runs through the membrane or the sarcolemma, making it more positive or more negative. And in that way, you're able to stimulate the cell. Our goal when we try to excite the sarcolemma is to make the membrane more positive, so make it more excitable. So part of that is going to be by opening up these ligand-regulated ion gates and allowing sodium to come in and potassium to come out. And really what's happening is that sodium is flooding in at a higher rate than the potassium is leaking out. But that's something we'll discuss at a future point. For now, it's just important to point out that these are ligand regulated. and they're going to cause a membrane potential to become more positive. So we're exciting the sarcolemma, and we're creating what we call an end plate potential. End plate potential. So we're exciting the sarcolemma, but it's very localized. It's very localized to where the receptors are. All right, so now take a look at this picture right here. So we have our axon terminal, right? We have our little synaptic cleft right over here. We have our sarcolemma. And then you see your little receptors, your acetylcholine receptors that we were just talking about. And remember how we said that they are ligand dependent. So when the acetylcholine attaches to it, it's going to open up the ion gate and it's going to allow for an end plate potential. So a very localized excitation to occur. Well, it turns out that that excitation is going to start spreading across the sarcolemma, away from the synaptic knob to the rest of the membrane or the plasma membrane of the muscle cell. And as it goes away from the axon knob or the neuromuscular junction, it's going to encounter what we call voltage regulated ion gates. Now these are going to be called voltage regulated because they're going to notice the change in the membrane potential. Remember how we're making it more positive, we're getting it all excited. So as the sarcolemma becomes excitable, its end plate potential starts to spread across its entire length and will eventually encounter these voltage regulated ion channels. And these ion channels will notice the change in membrane potential, and they will respond by opening up. And when they open up, it's going to allow sodium to go flooding in at a higher rate, potassium to go flooding out also at a higher rate, and this is going to allow for a large-scale action potential to occur. An action potential indicates that you have reached the point where the cell is so excitable because you have altered its membrane potential that it's ready to give you a response. In this case, our response is that we ultimately want the muscle cell to contract. But remember, the contractile units are inside the muscle cell. Right now, we're still on the sarcolemma, the outside portion of the muscle cell. So we need to figure out how to take all that excitement from the sarcolemma and move it into the actual cell so we can start taking a look at our thin and our thick filaments. In order to do that, we're going to move to our second step, excitation-contraction. So we are going to couple our excitement, so our changes in membrane voltage, to the actual contraction that we want to have, so the response that we want the muscle cell to have. In order to do that, we have our action potentials that we were able to generate. We're going to have to take that excitement and get it inside the cell. So what can we use to do that? Well, what about those little invaginations that we were quickly introduced to in part 1, the T tubules? The T tubules are little, almost like pathways that allow for an opening to occur in the sarcolemma that penetrates deep into the muscle cell. So notice here how you have these little yellow arrows. The yellow arrows are meant to represent my action potential that started off with the synaptic knob and then traveled through the sarcolemma, and when it comes across a T tubule, that action potential, that excitement, is going to be able to go from the outside sarcolemma to the inside of the muscle cell where I have my thin and my thick filaments. And as it starts to spread inside the cell, it turns out that the T tubules are surrounded by SR region. That stands for sarcoplastic reticulum. They're going to be very important because guess what they store? Calcium. So when the excitation travels down the T tubule, the sarcoplastic reticulum picks up on this and releases calcium. Now, why do we need that calcium? Think back to part 1. Think back to that relationship between your thin and your thick filament. And there's something that's blocking their interaction, something that requires calcium to get it resolved. Maybe the next slide will tell us what that is. Here you go. So once you have your action potentials coming down your T-tubules, once you have your calcium being released from the sarcoplastic reticulum, calcium starts flooding the inside of the muscle cell. Well, where's the calcium going? I hope that right now you're saying to the troponin, and you're completely correct in that. So remind yourself in part 1 how we talked about the fact that your thin filament is made out of actin, right? These little red structures right here, and they have a little active site. Look at these beautiful active sites right here. A little dip where the head of the myosin perfectly fits in. And they will only expose the active sites when you want the muscle to contract. If your muscle isn't contracting, it's covered up by tropomyosin, and tropomyosin is controlled by the troponin knob, and it will only react to calcium. So when calcium comes out of the sarcoplastic reticulum, it will travel over to the troponin. The troponin will move tropomyosin out of the way, and when it moves the tropomyosin out of the way, now we have exposure of the active sites. And because we have the striations in skeletal muscle, we know that we have alternating sections between the thick and the thin filament. So there is a thick filament very nearby, that thin filament, ready for its golf ball head to interact with the active site on the thin filament. So this brings us to the third step, which is the actual contraction of the muscle. In order to do that, we're going to need to go ahead and create what we call a cross bridge. And here it is highlighted as well. A cross bridge is going to be a physical connection between the head of the myosin and the active site on the actin. So they need to physically make a connection. In order to do that, we do require ATP. So you're going to need to take an ATP molecule, a chemical energy molecule. You're going to hydrolyze it, and it's going to cause the head of the myosin to go ahead and what they call cock or change its position, so it can go ahead and attach and make a physical connection. And that physical connection is a cross bridge. All right, so the cross bridge is one of the first things you need if you want to actually see a contraction. But remember, with a contraction, you need it to shift. You need it to slide. We can't just connect. So what we're going to see on our next step is this thing called a power stroke. Here we go. So once we made our initial cross bridge, we see that we're going to take another ATP molecule, and it's going to break some of the cross bridges so that the head of the myosin can go ahead and detach from its original location, and it's going to be able to glide over to a secondary location. So starting right here at the bottom, we're going to see a power stroke. And a power stroke literally means that the head is going to remove itself from its initial location, and it's going to go ahead and make a secondary attachment right here, and it's going to slide the thin filament along the way. So it's going to look a little like this. Here's the little illustration that you have, and you can see you have your thin filament indicated by green, and then in blue with those little circles, those are the head of the myosin. So what they're doing is this thing called power stroking. They basically release one section of the active sites, grab another one, and pull it through. And in fact, what you can do is you can come look over here on this side, on the bottom left, and you can see how you have your little calcium molecules that are coming in. And then notice how the head of the myosin makes a cross bridge. And then you see how it shifts it to the back. So here comes my calcium. It's going to attach to my troponin. It exposes the active site. We're going to do a cross bridge. So we're going to make that physical connection. And then we're going to slide. So give it a second. You see that sliding motion that it just did? That is the power stroke. And that requires ATP to be successful to do so. And as it's power stroking, it's basically overlapping the thin and the thick filament. So looking back at your top illustration, you can see how the Z lines get closer together. that then means that your muscle is contracting. And when the Z lines go back to their original position, that means that the muscle is relaxing. So the contraction is all about, one, making a cross bridge, a physical connection between the thick and the thin filament, and then doing a power stroke where the head of the myosin basically goes to another active site and allows the thin filament to slide over the thick filament, thereby creating an overlapping area, allowing the Z discs to get closer. And the sarcomere, your contractile unit, it shrinks, right? Because it gets closer. And that is then what we see as a muscle contraction. This is what they like to call the sliding filament theory. The fact that when you contract the muscle, it doesn't reduce the size of the muscle. We're simply overlapping the components. So they're sliding over each other. And this will continue as long as you have your nerve signal, as long as you have your calcium molecules, and of course, as long as you have your ATP molecules to do this. The last step is going to be, of course, relaxation. So once the muscle's job is done, we need to relax and go back to its resting length. So what we see happening is that the nerve stimulation will stop. The nerve signal will no longer come down the axon. We will no longer release acetylcholine. In fact, any acetylcholine that's still in the synaptic cleft, the acetylcholine esterase, here you go, that enzyme is going to come on down and break it down. It's no longer needed. Notice how it kind of looks like a little crescent moon. And when the acetylcholine stops being released, then you also stop interacting with the acetylcholine receptors, which means that you no longer have an end plate potential. You no longer have your voltage-gated ions that are opening up and creating an action potential and getting the membrane, the sarcolemma, all excited. So when the T tubules notice that there's no longer an action potential coming down, the sarcoplastic reticulum will respond by reabsorbing the calcium. So all the calcium that's still around floating inside of the muscle cell, it's going to get reabsorbed by the sarcoplastic reticulum. And once the calcium goes away, what we see happening is that that will be detected by the little troponin. Remember the little knob that's dependent on calcium to move tropomyosin? Well, if we remove the calcium and allow it to go back into the sarcoplastic reticulum, we see that the calcium, once lost, will trigger the troponin to move the tropomyosin back into its original location, thereby blocking the active sites. And when you block the active sites on the thin filament, then the myosin head can no longer make physical contact, can no longer make those cross bridges, and can no longer make the power stroke. So once again, you get rid of that connection between the thin and the thick filaments, so you stop sliding, and that this causes the Z discs to basically go back to their resting positions as it was, allowing for the muscle to relax. Also, what I want to point out is that it does require ATP for the muscle to relax. So you require chemical energy both for muscle contraction as well as for muscle relaxation. So please take your time, walk yourself through the steps, be able to explain all the 4 steps. So your excitation, your coupling, your contraction, and your relaxation. Please be able to explain the role that calcium plays in it, the end plate potential, what the ligand-gated receptor is, what the voltage-gated receptors are, all the different players like troponin, tropomyosin, why you need ATP for both contraction as well as relaxation. And of course, take your time going through this. And if you have any questions, you know I am available through our Blackboard Collaborate on a weekly basis, but you don't have to wait that long. Shoot me an email and I'll be more than happy to explain or set up a little Zoom meeting for us so that we can kind of discuss the materials. This is a very detail-oriented concept, So sometimes it does take a few tries to kind of see all the moving parts, to see how it kind of fits all together. To wrap up chapter 11, I want to talk to you about what we call the link-tension relationship. This is concentrated on what we like to call muscle tone. So at a resting location, how much overlap do you have between the thin filament and the thick filament? And the reason you want to study this is because you want to have what we call optimum resting length. And optimum resting length just indicates that at rest, there's going to be a slight amount of overlap between the thin and the thick. I'm going to put a little box around it right here. You can see that they're overlapping, but you're still going to have some empty sections on the thin filaments. The reason this is important is because, one, you want to have overlap so that when you want to form your cross bridges, the myosin head can easily connect with the active sites on the actin. And then you want to have some thin filaments that's left blank because, remember, you're going to do your power strokes. You're going to start gliding. So you want to have an area on your thin filament where you can glide to. And this will then allow you to have optimum amount of cross bridges as well as power stroking. So you'll have a nice muscle contraction. If you overstretch the resting length, then that means that the muscle is already extracted. It's minimizing the amount of cross bridges it can initially make. It's going to have a hard time making that initial contact between the thin and the thick filament. And if it does so, it has a hard time making the cross bridges. It's going to have an even harder time gliding and power stroking. This is what we see, for instance, when we overstretch a muscle. So for instance, when people say that you have to bend through your knees, not through your back. And the reason for that is because your back has this really thin bundle of muscles called the erector spinae muscle. And if you're standing and you want to kind of reach down to grab a box that's on the floor, when you bend your back, you start to overstretch those muscles. So now that muscle's already overstretched. So by the time you get to the box and you want to generate power for them to actually do their cross bridges and power stroke, because they're so overstretched, they have a really hard time making that initial connection. And this is then stimulated by the muscle starting to hurt and you being in pain and feeling overall weak, like you can't grab the box, but that's because you're not utilizing the correct muscle fibers. It's the same thing you see when the muscle is over contracted. This just basically means that if you're at the level where the muscle is already engaged to the fact that you have made the cross bridges and you have maximized your power stroke, meaning there are no empty spots for it to continue to slide, you cannot further contract that muscle. It has been over contracted. So please take a look at this graph, and once again, it's all about calculating the perfect optimum resting length. This is what we like to call muscle tone, so that when the muscle becomes engaged, it's easy for it to make cross bridges quickly, and it has room to do its power stroking. The last entity of part 2 is a clinical application. I wanted to discuss rigor mortis. Rigor mortis is basically when we see a person becoming deceased, and about 3 to 4 hours after that, we see that the muscles will harden and it will start stiffening the body. So this is often due to the fact that the minute the body becomes deceased, it no longer produces ATP molecules, and its internal organelles will quickly start to deteriorate, will start to fall apart. One of the first things that fall apart will be your SR region, and the sarcoplastic reticulum will then cause a massive amount of calcium to be released within the muscle cells. These, of course, will be detected by the troponin, and it will slide the tropomyosin out of the way, which will then quickly allow for a cross bridge to form. The problem at this point is that you have your cross bridge, your physical connection, but you can't do your power stroking all too long, and you definitely can't relax because there's no ATP. Your body stops producing ATP the second that you become deceased. And ATP, whatever is left over, is utilized rather quickly. So what we see happening is that the muscle will contract, and it kind of gets locked in that hardened position, because it doesn't have ATP to go into the relaxation step. Now, this is not a permanent condition. Usually around 12 hours after death, the rigor mortis will peak, meaning it will be at maximum for most of your muscles, and then it will slowly start to diminish because the actual fibers, so like your thin and your thick filaments, will start to deteriorate, and they will naturally get rid of the cross bridge because they're falling apart. And as the cross bridges fall apart, then so does muscle relaxation occur, because once again, there's nothing there to allow the myofilaments to interact. The sarcolemma, the contractile unit, the sarcomere, the contractile unit is literally disappearing. It literally falls apart. And what we see happening is that usually after about 48 hours, the rigor mortis will be completely gone. If you watch any of your investigative / detective show, you know that when they find a body, they can use rigor mortis as a way to kind of establish when that person became deceased. And usually if they find the person in rigor, they know that it's relatively been a short period of time since that person has died. Whereas if rigor mortis has already come and gone, then they know it's a longer period since the person became deceased. Alrighty. The good news is that this is the end of chapter 11, and I look forward to your questions about the content. And keep in mind that we do have our discussion sessions. You can find me through email or stop on by one of my Blackboard Collaborate sessions. Alrighty, so we will talk soon when we go into chapter 12.